I want to thank NHGRI for funding us all these years, and TDCC for managing our sometimes late reports, and WashU for having us here. Thank you. So 5 years ago, my wife and I had twins, and because my wife and I are both RNA people, we gave this book to the first book we gave to our kids. Okay, and if you know, this is the picture of my kids when they were a few months old. I asked my wife for permission, so this is okay to show the picture. But if you can see, already sort of, you know, presented a bit by Sterling that this RNA is, you know, it has eyes and eyebrows. Okay, but really it's single-stranded. It has structure, and I hope to tell you at the end of this that's not the simple picture of RNA. So we want to step back, right? So I sort of tailored this talk a little bit to educators as well. This is the old picture, right? DNA encodes genes. Genes can be transcribed into mRNA, and then you translate it into proteins, right? And so for a long time, and even when I was growing up, we had this model where one gene equals to one mRNA equals to one protein. And you could actually, you know, take the genome sizes of prokaryotes and viruses and just divide it by 1,000, and you get roughly the same, you know, roughly the number of protein coding genes. And so if you look at the table, right, well, from eons ago, you see viruses, E. coli, and so on. You can see that on the left column, number of protein coding genes, and then you just take the genome, divide by 1,000, it's pretty accurate. But this estimation went out the window once eukaryotic genome started being sequenced, right? So you can see from Cerevisiae, right, Baker's yeast, and so on, right? You can see that it starts diverging, right? And then you get to humans, you know, why do you only have, what, 5 times as many genes from E. coli, right? And so the estimation has gone off. So the question that many in the field wanted to address a long time ago is, well, how do these 21,000 human, you know, protein coding genes, and we don't even include the non-coding stuff, right, generate organismal complexity, hundreds of thousands of different proteins. So Sterling talked about splicing, right? And so you have to give credit to Rich Roberts and Phil Sharp for the discovery of splicing. And you can see the central dogma nicely depicted on the left here for bacterial genomes, right? So DNA, messenger RNA, and then protein. But for higher organisms, you have this process where non-coding regions between coding bits, which are exons, are spliced out. And for this discovery, you know, for split genes, we now understand, well, perhaps why the genome is so big, but you have only that many genes in there, right? Because a lot of it is taken up by non-coding space. But then how do you get this diversity of proteins, right? So when you look at splicing, you can have this picture where different exons can be included at different times. So for example, in this 5-exon containing gene, you have a pre-messenger RNA with 5 exons, but you can splice them in different ways to generate different proteins. And so if you look at this picture, then it's a different model, right? So one gene can encode many, many mRNAs, and from that you can encode many different proteins. I think some of the extremes, like in DSCAM in flies, are at 38,000 isoforms from one genetic loci. Okay, so then how is splicing controlled, right? Well, it turns out splicing is controlled and orchestrated by many, many RNA-binding proteins. So for example, you have U1, U2 complexes that bind the splice sites up there. You have SR proteins and HIV proteins that coordinate to enable exon inclusion or suppression. And then you have, you know, positional effects from RBPs, splicing factors that bind upstream or downstream of a alternately spliced exon. But this picture is even more complicated. And just to introduce to you that every single step of every single RNA processing event, non-coding, coding, small RNAs, circular RNAs, whatever, right? All orchestrated by RNA-binding proteins. And so a single RNA can interact with many RBPs throughout its life cycle step. But so keep in mind that many RBPs bind to multiple, in fact, hundreds to thousands of RNA substrates in a eukaryotic cell. So when I started my lab, there were maybe 500 annotated RNA-binding proteins in the human genome. And then in 2012, 2 papers, one from Matt Hens's lab and another from Marcus Lenthaler's lab, use UV cross-linking of RNA to protein, to all proteins in the cell. And that creates covalent interactions, so you can then pull down the poly A RNA and then mass spec all the proteins that bind these RNAs. And so it turns out that there are thousands of uncharacterized RNA binding proteins in the human genome. And then later on, many labs, including ours, followed up with similar studies looking at non-coding interactions, different fractions, and so on. And recently, it's estimated that about a fifth of the human proteome encodes RBPs, right? So that's about 4,000 plus. So in my opinion, RBP biology is a blue ocean, right? There are thousands of uncharacterized RBPs. So what are the multiple ways RBPs control RNA processing? Are they even important for health, aging? Are they disrupted in disease? And can we use these interactions for drug discovery? And so, as you can quickly see, many of them have no known function, even molecular function, right? So one of the things that we were interested in is, can we assign a molecular function to not just one? We don't do things one at a time, but thousands of RBPs, right? And so there are simple ways to do this. So one way I want to introduce is called tethering. So you can take an RNA binding protein, fuse it to a MS2 coat protein from bacteriophage, and these MCPs have evolved to bind MS2 aptamers, and you can stick this MS2 aptamers into reporter RNA, right? So in this specific case, we generated a reporter with an exon in the middle, exon 10, that can be included or excluded, right? So this is tailored so that if you tether this RBP to this MS2 loop downstream of the exon, if the RBP causes exon inclusion, then you see this isoform that's made. If it causes exon exclusion, then you just see this isoform that's made. And some clever design of Renilla and Firefly, you can measure this in some sort of luciferase ratio. You can compute percent spliced in, which is the percent inclusion of the exon. And so you can tether positive controls, right? So negative control, flag control doesn't change the isoform ratio, but positive controls, you cause exon inclusion. And of course, you can orthogonally test this out by RT-PCR and then running a gel, where you see one band, one isoform, two bands, both isoforms, but there's more inclusion, and then you can see how that correlates with the luciferase acid. So the nice thing about these tethering assays is that you can look at effectorability of the protein. Can it affect splicing, for example, right? Independent of its binding affinities, because you're forcing it there. You can look at direct effects versus some upstream effect when you knock it down somewhere, and who knows at what point does it actually modulate RNA. And you can look at position-dependent effects. So we had run this assay for many different kinds of reporters over the years. So our first paper was actually looking at which RBPs control RNA stability or translation by tethering it into the 3' UTR of different reporters. And then, of course, this is the exon inclusion one. And we have other papers that will be coming out showing which RBPs can affect alternative polydylation, circular RNA metabolism, and so on. But with all these RBPs, you can imagine that it's easy to mutate them or deplete them in diseases, and that results in things like myotonic dystrophy, ALS, Huntington's, Fragile X, glioblastoma, and so on. So we study many of these diseases here in bold. And we also care about their binding sites, because mutations in their binding sites can also cause human disease. Maybe a good example here is Adrian Krainer's work from Cold Spring Harbor on spinal muscular atrophy, right? So this is a severe motor neuron disease where you get systems-wide muscle wasting. And it's a disease caused by loss of function of SMN1, which encodes the SMN protein. Now, it turns out in humans, there's a parallel SMN2. And if you can include exon 7 of SMN2, then that functionally makes SMN protein rescuing the disease, right? And so Adrian was able to show with Ionis and then Biogen, right, that by blocking a single RNA binding protein binding site, HRMP-A1, that represses the exon inclusion, you can cure the disease. And this is one of the first, I think, neurodegenerative cures with an ASO. And so since since you are interested in these sites, how do we identify these important RNA RBP interactions? And so, for example, interested in this green RBP, you have many RBPs, 4,000 of these. How do you pick out the green, the RNAs that are bound by the green ones? Coding, non-coding, cytoplasmic, whatever, right? So building on an original protocol from Bob Darnell's lab, many labs, including ours over the years, have tried to scale and expand this protocol we call cross-linking and IP or CLIP. And we call this enhanced CLIP because it really enables very large-scale screening, identification of RNA binding protein RNA binding sites. So for those who are not familiar, we start with cells or tissue that you UV cross-link these protein RNA interactions to form covalent interactions. Then you trim away all the RNA that's not protected by the RBP, followed by pulling out the protein and then the protected RNA, ligating adapters, running it through gels to clean up the background, and then ligating adapters at the end for sequencing. And so the data sets we get are these eCLIP IP data sets where you have, for example, RBFOX2 and the RBP binding sites next to the RBFOX2 motif, UGC, UG. And it's important that we use input controls that are, in this case, very clean, so you can compute enrichments. And so we were excited to have the opportunity from NHGRI to map what then was the largest number of RBPs to date in basically one large experiment, right? So this is some of the lessons we've learned, right? So we found that each RBP has a mostly unique set of binding sites. It's a bit frustrating because that means that we've only mapped 150 RBPs. There's 3,000 plus more, right? But the binding sites are indicative of their function, right? So for example, here, DIN28 binds to coding regions in red, right? Just for ease, to control translation. RBFOX2 binds specific intronic regions. Every now and then, there's the UGC motifs, and so it binds the specific one. PRPF8 binds to 5' splice sites, so almost all 5' splice sites have PRPF8 binding. And U2F2 binds to 3' splice sites, and IGF2BP1, for example, binds to the 3' UTR to affect localization and stability and so on. And so we've been quite interested to first map all these sites, but that's not sufficient. You have to have orthogonal methods. So collaborating with Brent Gravely's group, Chris Birch, Songdong Fu, and Eric Luquier, we were able to then generate in vitro binding motifs for a large set of these RBPs, IF images for all of these ones that we have antibodies for. These are all antibodies that we can use for endogenous IPs, knockdowns, and then chip-seq as well. And so this, by the way, NHGRI was the largest binding and functional map of RBPs to date. We have actually doubled our eCLIP data sets, and we're trying to get this manuscripts out. Now we've continued to develop new technologies. So for example, the eCLIP method and other methods are great for lots of cells, but you can't really get to single cells with it. So we have developed STAMP, which is a fusion between an RBP and RNA editing enzyme. So wherever the RBP goes, the RNA enzyme then edits CDU, for example, here. And we've written the algorithms to identify these and so on. So this is the very first single cell level binding event for an RBP, so RBFOX2, right? So at single cell level resolution, you can see edits at each individual cells, whereas control is relatively clean. Now we have developed this so that you can do this in situ, so you don't even have to overexpress these. You can just float in an antibody and then float in a nanobody that recognizes the antibody. Now, RBP localization is actually not just static. So these binding maps depend on the state of the cell. So here in this case, RBPs are highly dynamic. So someone mentioned just now, I think if you stress cells, what happens? So this is GGBP1. And you can see in green, GGBP1 coalesces into these organelles, non-membrane containing organelles very quickly over minutes and then recovers. This is an important process, right? Because essentially every neurological disease, neurodegeneration disorder, contains RBPs that are part of these sort of condensates, and many of them are mislocalized. A good example, in ALS, which is a severe upper and lower motor neuron disease, essentially all patients have TDP-43, which is an RBP, mislocalization in the cytoplasm. So we all start with nuclear TDP-43, so present in all our neurons, and actually in all cells. And then at end stage in disease, for ALS patients, you see that they are in the cytoplasm and they're in aggregates. How does that happen? It turns out, we have a recent study that was published, I think, a few weeks ago, that this is not just in disease. If you look at old brains and or mid and then old cohorts, you already see that many RBPs, in fact, that's the strongest signal for neuronal aging, they're mislocalized or depleted. And this example here I'm showing is TDP-43 is already mislocalized in older brains, right? Wild-type individuals. So that gives you an idea why some of these diseases are age-dependent, right? Why the germline mutation in TDP-43 doesn't do anything until you get to some point in age where then there's a mislocalization event. Okay, I want to remind all of you though, that RBP-RNA interactions, we already know that they're important for therapeutics, right? Because antibiotics are small molecules that interrupt ribosome interactions with RNA, gene silencing events, siRNAs, ASOs. The business end is the RBP, right? Argonaut 2 or RNase H. And then obviously CRISPR is the same. The business end is a CRISPR protein, but guided there by the guide RNA. Okay, so in my group, we've also trying to engineer new RBPs from scratch. Can you build, we built machine learning algorithms to extract relevant RNA binding domains. Then we can, I'll show you in the next slide how we try to engineer them to bind different motifs from what they normally bind. And then we can equip effector proteins from our tethering screens to then effect RNA the way where we want to. And so this is a pilot experiment, right? Can you take one single RNA binding domain? We've collected experimental data to show that you can make rational mutations to change specificity from the RNA binding domain, the single small domain recognizing a GGU motif, to a GGG motif, because the hydrogen bonding between the uridine initially is now changed and actually preserved if you use an arginine and to a guanine. So this is preliminary data. It seems very small, but it argues that you can now systematically go through domains and engineer them. Okay, so I just want to leave you with this. RBP binding sites are abundant, they're learnable, and they're designable, right? And if you think about, from a small data set of 150 RBPs, we already estimate that a third of exonic sequences have a binding of one RBP, which means that if you think about 4,000, almost every single RNA is coded, right? Which brings us back to the takeaway. So RNA is never naked. It's always clothed by RNA binding proteins. Okay, so with that, thank you for your time and attention. And I want to thank funding sources, as well as my lab on the right, and then the other organizations I've been a part of. Thank you.
Wonderful. Thank you, Gene. Questions for Gene?
Okay, so I'm really interested about okay, so you said there's certain RPGs are for certain diseases. Is there a certain localization that they're specific to? Are they okay, so they're always on the blank? Are there always, is there a correlation?
So Eric Lucas' lab in Montreal has taken the antibodies that we have provided, I think, at least 1,000 antibodies, and he has labeled cells, individual RBPs, right? And if you look at this very beautiful image from Eric's work, right, they have very diverse localizations, right? Yeah. Individual ones can be in the nucleus, can be in speckles, can be in just the synaptosome in the neuron. So it turns out that when you look at the disease-specific ones, localization and cell type specific expression really matter. Okay. And that's how that's why we're trying to get to cell lines and tissues which represent that abnormal state, including age tissues, right? Yeah.
All right, thank you.
No, a really awesome message. The packaging of RNA is super important, and I'm wondering if there's any efforts to combine these RNA editing techniques with long-read sequencing to get co-occupancy data.
Yeah. So our STEM technology, which is on some slide somewhere, where we fuse the ApoBex to RBPs, right? We have in that initial Nature Methods paper show that you can do long-read sequencing to get isoform-specific binding, which you cannot get with CLIP because you digest away all the RNA. Here, you don't digest, right? You keep the isoforms. We can get ribosome occupancy also with isoform specificity, because ribosomes are also RBPs. You can fuse the ribosome subunits to ApoVec. And then Kate Mayer's lab has shown that you can take the other approach, which is TRIBE, which is ADAR editing, A-to-I, and then STEM, and then do them both on a single RNA to look at co-occupancy and single molecule level. So there's a lot of things that can be done. Yeah.